We will continue our discussion about this GPI-APs expression on the Calnexin and calreticulum dual knockout cells and how they differ from the GPI-APs that are expressed on the wild type cells. So in this experimental figures, that is panel, that is figure on the left hand side, they have analyzed that GPI-APs that are usually expressed in the wild type hex cells have wild type hex cells have this alkaline phosphate activity. They need to check whether these activities still exist in the GPI-APs that are expressed on the Calnexin and calreticulum dual knockout or not. So first they check the sensitivity of this GPI-APs expressed on the cell surface with the help of PI-PLC. So that will give them an idea whether there is a deacylation occurs or not. So in this case, what they have find out is that there is a partial, there is a partial sensitivity of the GPI-APs that are expressed in the dual knockout compared to the wild type, and these things are represented in quantitatively in the panel C, where we can see that the remaining of the ALPPs after PI PLC treatment is statistically significant, or it's higher compared to the wild type in the case of dual knockout, and is statistically significant. However, when they check the expression of this ALPP, they can see that there is no difference they have found out in the wild type and in the dual knockout GPI-APs that are expressed on the cell surface. But when they check for the activity of this ALP, they can see that the activity is statistically lower in the case of dual knockout compared to the wild type. So from these results, from these experimental results, they come to a conclusion that non-functional GPI-APs are expressed on the cell surface of this Calnexin calreticulum dual knockout. However, when it comes to the wild type, these are the functional GPI-APs that are expressed on the cell surface. So one of the reasons that they made because of the non-functional GPI-APs expressed on the cell surface is the low retention time of the GPI-APs in the case of dual knockout. So in order to validate that with increase in the higher retention time, the GPI-APs expressed on the dual knockout also will have more activity. So what they used a construct, VFGPI construct that is modified in a way or mutated in a way that it will correctly fold it at 32 degree, however in misfolded occurs at 40 degree. So here the deacylation spelling is wrong, so it is DEACYLATION. It's a typo error. So here what they did in this experiment is that they grow the cells at 32 degree and also at a 42 degree. Panel A figure shows the fluorescence image of 42 degree, at 40 degree, where we can see that the overlap of VGF, GPI and KDLs in the wild type samples is seen predominantly correct. However, in the case of wild type samples, we can see that this VGF GPI is not only expressed in the endoplasmic reticulum but also on the cell surface, as we can see that the signals are not aligned at several places compared to the cadence. And when they check it for dual knockout, they can see that the signals are overlapping. So this indicates that in the dual knockout, the misfolded proteins remains within the ER, and with the dual knockout and with the transfection or with the rescue of this Calnexin wild type, they can see that the more GPI-APs are expressed on the cell surface. So this is evident because the Calnexin and calreticulum helps in the properly folding of the GPI-APs, and these things they are validated using the panel B quantitative analysis is shown in the panel B, where the results not only shown for the 40 degree but also for the 32 degrees. For 32 degrees, they can see that there is a In the wild type, they can see that the PI-PLC sensitivity of the GPI-APs are higher, and for the dual knockout, it is partially sensitive. However, for when it is rescued with the Calnexin wild type, the sensitivity of PI-PLC treatment to the GPI-APs increases, and similar results they have seen for when the cells are treated at 40 degree, and after that 32 degree for 1 hour. The results are similar that can be seen here. However, for this, except for the dual knockout also they can see that sensitivity increases like around by 3 to 4 fold compared to the case where cells are grown in 32 degree. So at 40 degree, the more GPI-APs are expressed on the cell surface, and they are more sensitive towards this PI-PLC. So this one reason could be that at 32 degree, what will happen is that the cells in the at 42 degrees, since the proteins are now misfolded, they will retain in the ER for a longer duration time, and when it turns to 32 degree, and they will have the ability to move to the plasma membrane because of the acquiring correct folded structures. So now they need to check whether this cell surface GPI-APs are are sensitive to the deacylations or not. So in this figure they show that they are more sensitive to a deacylation compared to the case where this VGF is cultured at 32 degree for 24 hours. And results are quantitatively analyzed in this panel C, where we can see that when the cells are grown at 32 degree, that most the GPI-APs on the cell surface are less sensitive to the PI-PLC in the case of dual knockout. However, when it compared to the case of wild type and the dual knockout in which Calnexin is rescued, Calnexin function is rescued. However, when the cells are treated at 40 degree for 24 hours and then 32 degree for 1 hour, they can see that the GPI-AP sector on the cell surface are non-significant in all the three cases. So this indicates that with increasing the retention time of these GPI-APs in the cell surface will help in the deacylations, improving the deacylation properties of them, or deacylation properties of GPI-APs. So these 2 points are being validated in this figure, that is non-functional GPI-APs are expressed in the dual knockout, that is Calnexin and calreticulum both are knocked out in the cells, and with increasing the retention time their functional properties could be increased, such as deacylation of GPI-APs. And again, reminder is that the deacylations is misspelled in this video, in this figure. So I hope you understood what are the different variation that occurs in the dual knockout, that is non-functional GPI-APs expression, and at the same time this non-functionality will be reduced if somehow the retention time within the GPI-APs in the entoplasmic reticulum increased. And if you need to understand more about these experimental results in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.